Hello everyone. So today let us try and understand the classification of lipids. Okay. So first, what are lipids? Okay, lipids are nothing but these are compounds which are insoluble in water, but they are soluble in organic solvents. Okay, like chloroform or ether. So we all know that lipid, if you try to mix the oil with water, it is not going to mix. Okay, so lipids are compounds which are insoluble in water, but they are soluble in organic solvents, non-polar organic solvents like chloroform and ether. Okay, so how are lipids classified? Let us look at the classification of lipids. So lipids are basically classified into four different types. The first one is simple lipids, then compound lipids, derived lipids, and lipids complexed to other compounds. Okay, so what are simple lipids? Simple lipids are these are esters of fatty acids with various alcohols. Okay, so simple lipids are having fatty acids which are in bond formation or ester formation with various alcohols. Now, compound lipids are these are esters of fatty acid and glycerol, plus it has an additional group which is known as a prosthetic group. Okay, now this group can be a phosphoric acid, carbohydrate, etc. Okay, so compound lipids are having fatty acid, glycerol plus an additional group which is called a prosthetic group. Okay, simple lipids are only fatty acid and various alcohols. Okay. Now derived lipids, these are compounds which are derived from lipids or precursors of lipids. Okay, so these are derived lipids are compounds which are derived from lipids. Okay, breakdown of lipids also is a type of derived lipids. Okay, and the last one is lipids which are complexed with other compounds. Okay, for example, if lipid forms a complex with proteins, it is called proteolipids, or lipids, or it can be a lipoprotein. Okay, so let us first try and understand this classification. Now, simple lipids. Okay, simple lipids can be either fats or oils. Okay, so these are esters of fatty acid and alcohol, which is glycerol. Okay, now if it is a solid form, okay, it is called a triglyceride, or if it is a liquid form, it's known as oil. Okay, so this is the structure of a triglyceride. Okay, that is, it is consists of an alcohol which is glycerol. So glycerol has 3 hydroxyl groups. To each of the hydroxyl group, you have a fatty acid which is attached. Okay, same over here, 3 hydroxyl group alcohol. Okay, so over here it was OH, again here OH, here again OH. So the H is replaced by a fatty acid. Okay, C double bond O. Okay, here also this is replaced by a fatty acid, and here also it is replaced by a fatty acid. So a glycerol consists of 3 hydroxyl groups, and it is replaced by a fatty acid on each of the 3 groups. So this is an example of triglycerides or triacyl glycerol. Okay, so triacyl, 3 fatty acids, triacyl glycerol. Okay, now if this if it is a fat, if it is a solid form, it is called triglycerides. Okay, if it is a liquid form, it is called oil. Okay, so that is the difference between fats and oils. Now, what are waxes? Waxes, these are esters of fatty acid with high molecular weight monohydric alcohol. Now, instead of this glycerol, if there is a high molecular weight monohydric alcohol, okay, then it is known as waxes. Okay, so glycerol has 3 hydroxy groups. In case of waxes, there is only 1 hydroxy group. Okay, so these are the examples of simple lipids: fats and oils and waxes. Okay. Now let us look at compound lipids. So we have now seen that compound lipids are lipids which have an additional group which is known as a prosthetic group. Okay, so these are esters of fatty acid and contain an additional group. Now based on this additional group, the additional group can be either a phosphoric acid, then it is called phospholipids, or it can be a non-phosphorylated lipid. Okay, that means a group other than the phosphoric acid. Okay. So again, this compound lipid is further divided into phospholipids. Okay, that means having phosphoric acid And this phosphoric acid can either have the alcohol, if it's having an alcohol, it can have glycerol, or it can have an alcohol known as sphingosine. Okay, whereas non-phosphorylated lipids, which do not contain phosphoric acid, they have alcohol, which is sphingosine. Okay, so what are these? That we will see. Okay, so compound lipids again divided into two types, one containing the phosphoric acid, which is phospholipid, the other which is not containing phosphoric acid, it is called non-phosphorylated lipids. Okay. Now phosphoric acid, again having the alcohol glycerol, and again long chain amino acid, long chain alcohol. Okay, and another type of alcohol which is sphingosine. So phosphoric acid, if it has glycerol, it is called glycerophosphatides. Okay, if it has another alcohol which is called sphingosine, it is called phosphosphingosides. Okay. And non-phosphorylated lipid, here the alcohol is sphingosine. Okay, so this is further divided into glycosphingolipids, that means having additional group of carbohydrates, and the same carbohydrate, if it also contains the sulfate group, it is called sulfolipids. Okay, so let us try and understand the classification of phospholipids. Okay. Now, this is an example of a phosphoric acid. Okay, so this is our trihydroxy group alcohol or a glycerol. Okay, so 1, 2, and 3. Okay, so here the hydroxy group is replaced by one fatty acid. Here the hydroxy group is replaced by another fatty acid, and here on the third fatty acid, it is replaced by, it is also having a phosphoric acid. Okay, phosphate group is attached, hence it is also having a, it is known as a phosphoric acid. Okay, so this is the basic structure of a phosphoric acid. So phospholipids having this phosphoric acids are classified as phospholipids. Okay, now let us try and understand the first one. Okay, nitrogen containing glycerophosphatides. Okay, now see over here, the example is lecithin. Okay, lecithin is phosphatidylcholine. So same thing over here, this is our alcohol. Okay, so what is the alcohol present in phospholipid? That is glycerol. Okay, so over here is glycerophosphatide, that means the alcohol which is present in glycerol. The first amino acid, second amino acid, and here we have our phosphoric acid. To this phosphoric acid, you have a choline group which is attached and which contains nitrogen. Okay, so phospholipids are again based on the alcohol. Okay, it is glycerophosphatides, and based on whether you have nitrogen containing or a non-nitrogen containing. So it's a nitrogen containing glycerophosphatide. Okay, example is lecithin, which is also known as phosphatidylcholine. Okay, similarly we also have is cephalin and phosphatidylserine, which are also examples of nitrogen containing glycerophosphatide. Okay, now let us look at non-nitrogen containing glycerophosphatide. So again, here with alcohol which we have is glycerol. Okay, here phosphoric acid, so it's a glycerophosphatide. Okay, having alcohol glycerol and phosphoric acid. Now here if you see over here, this is the group which is attached is inositol. Here you do not have a nitrogen group. Okay, so it is a non-nitrogen glycerophosphatide. The example is phosphatidylinositol. Okay, phosphatidylinositol. Okay, similarly you have is phosphatidylglycerol and cardiolipin, which is diphosphatidylglycerol. Okay, these are examples of non-nitrogen containing glycerophosphatides. Now let us look at plasmalogen. Okay, now plasmalogen is what? Plasmalogen is having a long chain alcohol. Okay, this is also a type of glycerol. Okay, here, okay, this is our same old glycerol structure. The first hydroxy group, second and third one. Okay, so we know the second has the fatty acid, the third one has a phosphoric group, that is why it is a phospholipid. Okay, now here in the first hydroxy group, it has a long chain unsaturated alcohol. Okay, in case of plasmalogen, in the first hydroxy group is attached a long chain unsaturated alcohol. Okay, and this is known as plasmalogen. Okay, so plasmalogen it has a ethanolamine group. Okay, so it is an example of ethanolamine plasmalogen. Okay, so ethanolamine group is attached to a plasmalogen, which is nothing else but a glycerophospholipid to which a long chain unsaturated alcohol is attached. So it becomes a plasmalogen. Okay, so this is an example of plasmalogen having long chain alcohol. Example is ethanolamine plasmalogen. Now the last one is sphingomyelin. Okay, here we have the alcohol which is sphingosine. Okay, so this is the alcohol which is sphingosine. Okay, so this is the sphingosine. It's a type of alcohol. It's an to which a fatty group is attached. So it's an amino alcohol. Okay, you have a amino group here to which a fatty acid is also attached, and you have a phosphoric acid. Okay, so it becomes a phosphosphingolipid. Okay, phosphosphingolipid. Okay, and we have a choline group which is attached, so it becomes a sphingomyelin. Okay, so sphingomyelin is a type of phospholipid which contains the alcohol sphingosine. Okay, now let us look at non-phosphorylated lipid. Okay, so here the non-phosphorylated does not contain the phosphoric acid. So here what happens is, I told you the alcohol which is present is known as sphingosine. Okay, now sphingosine is amino alcohol as we have seen. Now this is the sphingosine having a amino group which is known as an amino alcohol. To this amino alcohol, if a fatty acid is attached, it is known as ceramide. Okay, to this, if it is a fatty acid is attached, it is known as ceramide. And depending upon the attachment to ceramide, we have the classification of non-phosphorylated lipids. Okay, now see, same thing. This is a sphingosine. This is our long chain fatty acid which is attached. So sphingosine plus fatty acid gives you a ceramide. To this ceramide, if the carbohydrate is attached, then it becomes a glycolipid. Okay, and depending upon which type of carbohydrate is attached, the classification is given. If it contains a monocarbohydrate, okay, then it is known as cerebroside. If you have a oligosaccharide, okay, then it becomes a globoside. If the carbohydrate is also having, if it is an oligosaccharide and it also has a neraminic acid attached to it, then it becomes a ganglioside. Okay, so depending upon which type of carbohydrate is added to ceramide. Okay, so sphingosine plus fatty acid is a ceramide. So which type of carbohydrate is attached to the ceramide, the classification of glycosphingolipid is there. Okay, if it is a monosaccharide, okay, it becomes a cerebroside. If it is an oligosaccharide, okay, then it becomes a globoside. And if it is an oligosaccharide plus N-acetyl uramic acid, it becomes a ganglioside. And similarly, if the same thing, it also has a sulfate group attached, then it becomes a sulfated cerebroside. Okay, if a sulfate group is attached to a globoside, it becomes sulfated globoside. And similarly, sulfated ganglioside. So this is how the lipids are classified, okay, as phospholipids are classified. Next, coming to derived lipids. So I told you derived lipids are basically derived form of lipids. That means it can form a breakdown product of lipid also is there. So these includes fatty acid and glycerol. Okay, so we have seen simple lipid is made up of fatty acid plus glycerol, that is triglyceride. So if you break down this triglyceride, it forms fatty acid and glycerol. So it becomes an example of derived lipid. Similarly, steroids are formed from lipids. So they also come under derived lipids. Okay. Similarly, other products which are formed from lipids are prostaglandin, leukotrienes, etc. Okay. So we'll be studying all these in detail in subsequent lectures. Then coming to lipids complexed with other compounds. Okay. So it can be, when it combines with proteins, it can be either a proteolipid or a lipoprotein. Okay. So again, what are these? that we will see later. Now, what are the functions of lipids? Now we've seen the classification. Let's briefly look at what are the functions of lipids, okay, which we will again discuss in subsequent lectures. Now, lipids have variety of functions, lot of functions. We all know it's a storage form of energy. Okay, so triglyceride, which is stored in our adipose tissue, is a storage form of energy. It gives us energy under conditions of requirement, okay, when it is not met by the carbohydrates. So under conditions of starvation, okay, so that it forms as a storage form of energy. Now lipids are components of biomembranes. Okay, all cell membranes are made up of phospholipid, okay, which is again a type of lipid, okay, compound lipid that we have seen. So all biomembranes contain lipids, okay, and what is the clinical implication of that? That we will see later. Lipids also act as metabolic regulators, like steroid hormones, prostaglandins. Okay, so we have seen under derived lipids. So from derived lipids, you have formation of steroids and prostaglandins and leukotrienes, which can act as metabolic regulators. Okay, they regulate the various metabolic products. Okay, that again we will see when you talk about hormones and prostaglandins. Now lipids also help in absorption of fat soluble vitamins. Okay, so we have fat soluble vitamins, as the name suggests. Okay, they are soluble in fat. So they are soluble in fat, so they can be absorbed in our body. Okay, so what are those vitamins? Vitamin A, D, E and K. Okay, so these vitamins, they require fat for their absorption. Okay. Lipids also act as surfactants and emulsifier. Okay, lipids play a very important role as lung surfactants. Okay, they help the lungs to expand. Okay, and emulsifier. Again, lipid helps in emulsification of fat. So fat absorption also takes place. Okay, and the fat soluble vitamins are also easily absorbed. Then lipids, they also act as electrical insulators in the nerve fibers. Our neurons are coated with lipids. Okay, so that it acts as an electric insulator. Okay, there is no discharge of impulses to nearby neurons. Okay, it acts as a insulator. It also provides us insulation against external environment and protects our internal organs. Okay, so we have is white adipose tissue and brown adipose tissue. Okay, again the adipose tissue also, in case of especially in newborns, they help to generate heat. Okay, it protects us against external environment. And also the lipids are surrounded, we have visceral fat, so the lipids surrounds the internal organs also, so it protects the internal organs also. We all know that lipid subcutaneous fat also gives us shape. Okay, and it also improves the taste of the food, makes the food more palatable. So these are the general functions of lipids, which we will discuss in subsequent lectures. Now another very important question: what are the various diseases which are associated with lipid abnormalities? Okay, we all know lipid is associated with lot of diseases. Okay, so what are these diseases that we will study? So these disorders are, one is diabetes mellitus. Okay, so you all know that excess of lipid is there. If there is obesity, okay, that can be a risk factor for diabetes mellitus. How? That we will see in subsequent lectures. Again, lipids can get deposited in the arteries, okay, and can lead to atherosclerosis. It can cause ischemic heart disease or heart attack. Similarly, it can cause cerebrovascular disease or peripheral vascular disease. Okay, it can obstruct the blood flow to the brain or to the peripheral organs and result in peripheral vascular disease. Okay, if excess of lipid is there in our body, it can lead to obesity. Okay, and obesity comes with its own problems, so that we will see again. And lipids also is derangement of lipids also is seen in metabolic syndrome. Okay, metabolic syndrome again acts as a risk factors for various other disorders. So these are the very important disorders which are associated with lipid abnormalities. Okay, so what are these disorders and how does lipid affect these disorders? That we will see in subsequent lectures. Okay, so thank you very much. Hope you have understood the classification of lipids.